to you.
Thank you. Thank you, Sybil, and thank you all the NMF students and all the members of LSU who decided to attend this great presentation. I met Dr. Chappell a few years ago when I was trying to put together a project on COVID very early on in the early stages of COVID. And Dr. Chappell had joined us from getting his PhD and his postdoctoral training at Rice University in Houston and then at MD Anderson, where he trained under some very famous faculty members who have developed very novel approaches to clinical trials. He was the one who told me that there was something that was called adaptive clinical trial design. And I said, what the heck is that? And I think in my career, the demonstration of somebody that really knows their stuff is they can take something very complex and make it into a simple story. And believe it or not, within 30 minutes, I got an idea of what these adaptive clinical trial design was about. So not that I understood the intricacies of it, I just got a pretty good idea. And I thought it was a very, very interesting way of being able to conserve in terms of effort, conserve in terms of numbers of patients involved in the trials, and very rapidly start drawing conclusions to whatever the trial was wanting to answer. So since his expertise is so intimately involved in clinical trials, I thought this would be a great opportunity for our NMF students to hear about these novel designs, which are rapidly making inroads into our everyday clinical trials. So without further ado, Dr. Chappell has all kinds of accolades. He is a professor, an assistant professor in biostatistics in the School of Public Health, and is a member of our cancer center. So without further ado, I'm just going to transfer it over to him. Andrew.
Thank you, Dr. Choa. So yeah, my talk today is going to be modern statistical, because I'm a statistician, perspectives in clinical trials. And I want to just kind of give some intuition on how these things work with maybe some simple statistical methods that y'all could be familiar with. And then thinking about kind of the things that are out there. So I'll go ahead and get started. So this is, why I'm giving this talk, some of my background. So I'm an assistant professor in biostats here at LSU Public Health. I was at MD Anderson under Peter Thal from 2015 to 2018. And while I got my PhD in statistics at Rice University, we actually published two adaptive clinical trial designs together. One of them actually won Best Paper in Biometrics in 2019, which is one of the top statistical journals out there. So that's my, I guess, big claim to fame there. Also, I am a avid traveler. It's me and my wife at Florence in the spring. And I am a fur dad of obviously very Greek statistical names. If I would have known we were going with this trend, Zeus would have been named something else, but Beta is a common thing in statistics. Deltas are as well, which is my dog. My advisor on the bottom right is Peter Thal. He's at MD Anderson. He's been there since like 1991, I believe. And he was involved in, some of you might be familiar with the Simon design. So he worked with Richard Simon. They published some papers. He established phase 1-2 trials, has lots of citations, and he runs a lot of trials over there at MD Anderson. So everything that I've learned, I learned from him. He's a very good advisor, very, I guess, involved. He had my cell phone number and called me on weekends and things like that. But because of that, I have a lot of expertise about how to think about these things. And I'd like to, in this talk, give everybody some intuition about how these things work, explain some of the areas that they can be used, and how the future might look as far as adaptive designs. Okay, so here's the contents here. Again, I'm going to start with just a simple randomized experiment of efficacy. So tumor shrinkage. Does the tumor shrink? Yes, no. I'm going to talk about what is power in a clinical trial, because I think probably a lot of people are familiar with, okay, I need a power calculation or I need a sample size justification, but what does that really mean? So I'm going to explain that in the prospect of replicated experiments. Then I'm going to talk about an adaptive design. So it's going to be an adaptive design in the context of numbers 1 and 2. So a simple randomized experiment of a binary outcome with adaptive decision making. I'm going to talk about data-driven decision-making in the future, which is going on in a lot of other industries right now. Some cancer adaptive trial designs, and then I'll plug my my biometrics paper, phase 1, 2, 3 design, and just kind of thinking about how this could move, how you could streamline the process between testing drugs, even in mice models, moving them into early patient studies, moving that on to trials against the standard, and then getting these treatments out to patients that end up helping them. Okay, so simple randomized experiment of efficacy. So let's consider a situation where we've got a new treatment A, and it's tested against a standard treatment therapy B. So these are just notations, right? The new treatment could be, whatever. It could be, if we're talking about cancer, some kind of chemotherapy, and we're comparing it against another standard chemotherapy in that region. The goal here is to determine whether the treatment A improves tumor shrinkage, yes, no, compared to a standard treatment B. So you could imagine, right? If you treat a bunch of patients and you follow them in the future and you observe, yes, no, did their tumor shrink? And then you can look at proportions and see, okay, did treatment A actually increase the rate of tumor shrinkage compared to the standard therapy? So before we kind of jump into how you test this, right, we'd have some previous evidence that treatment A is safe and some potential observational studies. So we're talking about a randomized trial here, or at least a trial that's guided through statistical models. It's a prospective trial. Okay, so in a randomized control trial, in the setting, you'd establish your efficacy endpoint. So again, it could be like remission within 3 months, no progression within 6 months, or survival in 1 year, or even progression-free survival in 1 year, however you want to do it. Then you'll plan out your sample size, what statistical methods you're actually going to be using, and your protocol. So are you going to do blinding? How feasible is different decisions for that, the cost. And what you'll do is you'll go ahead and start randomizing patients between the treatments, treatment A and B, observe their outcomes, look at whether or not their tumor shrunk or whether or not they had remission, whatever your efficacy endpoint is. And then you'll use statistical methods to determine, is the treatment actually better? Notorious, right? So you're probably familiar with p-values, right? So that would be based on a p-value, the standard rule of thumb, right? Check whether it's less than 0.05. And if it is, then you can, suggest that treatment A is better. Okay, so I'm going to have a little bit of math here. I just want to kind of explain exactly what we're doing as far as like population quantities. So what we have here is pi A is going to denote a true probability of efficacy with treatment A, and pi B is going to be a true probability of efficacy with treatment B. We don't actually know what these are. Like, God is not going to tell me, hey, look, if you do this new chemotherapy, you're going to improve remission rates by 10%. I don't know what this is, but I can go out and collect random samples to estimate these things, right? Just a simple proportion. What proportion of people actually had an efficacy endpoint in both of these treatment groups? Statistically, our goal is to test the population rates of efficacy are equal. That would be a null hypothesis versus an alternative that they're non-equal. Okay, and so if you're familiar with statistics, you get a p-value for testing this. If your p-value is less than 0.05, that's the general kind of rule of thumb. You'll reject the null in favor of the alternative, and you'll conclude that one treatment is better than the other. So again, we have these things pi A and pi B. These are true efficacy proportions in each of these groups. We don't actually know this, but we can go collect data and try to estimate this or test whether these true proportions are equal. And I want to make sure that everybody has that distinction, right? So there's some kind of true thing always in statistics, always in what we're doing experiments for. There's some kind of true thing we're trying to get at. We don't know what that thing is, but we want to collect data to try and get at what these true parameters are. So you might be familiar with a two-sample test of proportions. If you've taken, it might be a long time, but if you've taken a stats class, you definitely talk about that in here. So just some little notation just to remind you, PA and PB, these would be the efficacy proportions in the two groups. So for example, here I have a thing, PA is 0.5, PB is 0.3. So that means the efficacy rate in one group, the people that got treatment A had a 50% response rate. And in the standard therapy, they only had a 30% response rate. So that's definitely an increase, right? But is it statistically significant? Notation wise, continuing that, NA and NB, these are the sample sizes, and I'll use these throughout the next couple slides. So for example, NA equals NB equals 100, which means 100 people in each treatment group. And out of those 100 people in each treatment group, I had 50 in one group that had efficacy and 30 in the other group that had efficacy. P here, this is the overall efficacy rate, excluding consideration of treatment A and B. Here it's 0.4. So it's just the average, right, of these two, since they have the same sample size. And you might remember this kind of formula here. So this is a formula. This is a z-test of proportions. Again, if you recall, definitely, if you've taken a stats class recently, you'll remember this kind of thing here. So this is one of the more basic tests that we use, which is why I'm using it to motivate adaptive trial designs. So this test statistic here follows a normal distribution if the null is true. You might remember looking at z-tables and finding p-values from there, whatever, right? From a statistical software perspective, you'll get the test statistic and you'll get a p-value. You don't necessarily have to go look at these tables. And in fact, I think progress would be a lot slower if we still had to do that. So we get a p-value, and if that p-value is less than 0.05, then we'll reject the null hypothesis. The null hypothesis is that pi A equals pi B, which means that the true efficacy rates are the same in each group, in favor of the alternative hypothesis, which is pi A is not equal to pi B. So the true efficacy rates actually differ between the two treatment groups, and possibly in the direction of A being better than B. So if I rejected the null and I saw, right, that these were the efficacy response rates, I would probably say, hey, treatment A is better, had a higher response rate, significantly better. Okay, so this is just, we're collecting a data, we're collecting a sample, we get a p-value, and we're testing this hypothesis. Now I want to explain what is power in a clinical trial. So if you've done a lot of research, you've probably had cases where, for example, you see in the literature that somebody ran an experiment and they had a small p-value, and maybe you've tried to replicate that experiment and your p-value was 0.15. That doesn't necessarily mean that the treatments actually aren't different in terms of their patient benefit impact. That could be just a aspect of randomization and the patients that you're picking, what your sample is. So this is going to kind of lead into power in a clinical trial perspective. So power and type 1 error probabilities, they're theoretical quantities. Okay, so they rely on random sampling of your data and you're from a population. So for example, in the experiment we ran before, okay, I got 100 patients here in treatment A and 100 patients in treatment B. If I got 200 different patients from somewhere else in the state, or I just, had that day 200 other patients that wanted to enroll in the trial, I might get different outcomes. So I have a different sample, I might have different efficacy response rates, even if treatment A is better. So from a theoretical perspective, right, if I ran an experiment thousands of times, so I went out and I did this experiment, and then I went to Mississippi, I did this experiment in Mississippi, then I went to Alabama, I did this experiment there, then I went all around the states, and I can even come back here and do the experiment again. If reality, there's no difference in efficacy, there will be sometimes, right, that I reject this null. Sometimes out of those many experiments. So I ran 1,000 hypothetical experiments, and if in reality, treatments A and B are not different in terms of efficacy, sometimes I'm going to reject the null and conclude one treatment is better. That's a type 1 error. Also, if I knew in reality that one treatment is better by about 20%, I want to reject the null in that case. So out of 1,000 trials, I'm not going to reject that null every time, even though it actually is better by about 20%. So how often do I correctly reject or not reject the null hypothesis if I know the truth? Now, again, I don't know the truth. God's not telling me, hey, treatment A is better. But from a theoretical perspective in statistics, you can assume that you know the truth, do simulations, and see how likely you are to make the right decision based on those truths. So type 1 error. Type 1 error in a clinical trial, again, what if in reality pi A equals pi B equals 0.5? So there's no difference as far as efficacy rates. 50% efficacy rate, whether or not you get treatment A or B. It's still totally possible to run the trial and you'll reject the null that pi A equals pi B. It's totally possible just from random sampling. And when that's true and you reject the null, that's a type 1 error. So that's that type 1 error probability you probably hear a lot about. So I give a simulated example here down here. As an example, let's say you have two different sample sizes, NA equals NB equals 10, which means 10 patients in each group. And in reality, you observe in your experiment, you observe for treatment A, an efficacy rate less than 0.7 and a efficacy rate in the B group of bigger than 0.3. You can actually reject the null in this case. You would totally reject the null hypothesis, and this p-value would be 0.024. So this happens with a probability 1.6% when the null is true. So this is based on simulations. So 1.6% of your experiments that you run, you get the wrong answer. Okay, and that's something we want to avoid. So thinking about type 1 error, we want to avoid saying treatment A is better than treatment B, because that's going to lead to more trials. That's going to lead to clinicians thinking that treatment A is better than the standard, potentially mistreating patients. We want to avoid type 1 errors. And from a pharmaceutical perspective, they want to avoid type 1 errors because they're going to go on and spend a lot of money developing this drug. Type 2 error. So type 2 error, imagine that if in reality, the efficacy rate in treatment A is 50% and the efficacy rate in treatment B is 30%. So treatment A is better. We know this. Somebody tells me it. It's still totally possible to run the trial and not reject the null. It's totally possible. And that could just be by random chance. So even though treatment A is better, somehow maybe it works out that their individual outcomes are about the same as far as efficacy rates. That's a type 2 error. So imagine here, I have some more hypothetical quantiles. Imagine we have sample sizes of 10 in each group, and we observe a 0.7% efficacy rate in one group or 0.8. And in the B group, we observe a 0.3 efficacy rate. In this case, we wouldn't reject the null. We get a p-value of 0.17 and 0.07 respectively, whether the first group has a 0.7 or 0.8 efficacy rate. This actually happens often in this setting with this small sample size. It happens about 95% of simulated trials. So this is not a trial design that you'd want. You're setting yourself up for failure. If instead you have a bigger sample size, so 100 in each group, then you only have a 20% probability of this happening. So what does that mean, right? If you run this experiment thousands of times, and in reality, treatment A is better, only 20% of the time you won't reject the null, which means 80% of the time you will, and you'll say that treatment A is better. Now, if you increase your sample size, you should be able to get that even higher. But this 80% here is what power actually is. So power, you have to set up in some kind of reasonable truth. So here's 50% and 30%. You do simulations to see how often you'll actually reject that null if these assumptions are true. And the proportion of times that you reject the null correctly is your power in this case. So again, power, that's the probability of rejecting reasonably chosen true alternatives with the experimental procedure provided. So type 1 error probability is on the opposite side. So that's if the null is true, so there's no difference in treatments. What's your probability across thousands of replicated experiments of incorrectly rejecting the null? So your goal here for setting up a study in general is you want to maximize power under reasonable truths. So here I have an example, right? If you got this new treatment, hey, I've got this new chemotherapy, I think it's going to improve tumor response rate. And you're rare, like a disease that's hard to treat, that historically has a 10% response rate. It's totally not reasonable to assume that your new treatment is going to have a 90% response rate. That's an 80% increase in response. It's totally not a reasonable assumption here. If you did assume that, you would need a much smaller sample size to have good power. But that's basically saying you have a miracle cure for this cancer. So you have to think about, well, what's reasonable based on the literature, what's reasonable based on mice models, things like that. And you want to power your study under something that's a reasonable improvement to actually have a high chance of detecting that improvement, while at the same time minimizing the type 1 error probability in cases that match research. So, for example, if I'm looking at a historic sort of efficacy rate around 50% for some drug and some standard, some different cancer type, I'm not going to be assuming that the standard therapy has a 10% response rate or 90% response rate. I'm going to be assuming it has maybe a 40%, 45%, 50%, 55%, 60% response rate, something like that. It's something reasonable based on what's observed. So considerations here in this: what's your sample size? Is it balanced or imbalanced? So you might have seen, right, studies where they randomize 2 to 1 in the news treatment arm versus the standard. That's totally fine, but those decisions actually do affect your power and your type 1 error when you go make decisions at the end of the trial. So if you do simulation studies before your trial starts, you can get an idea of how many patients you should enroll in each group, whether or not 2 to 1 or a 3 to 1 strategy is actually good, and other things like that. Okay, so I've kind of been talking about like hypothetical experiments. So imagine, now we've got Marvel, right? So imagine instead, right, you're running the same experiment and you've got all these multiverses, and in the different multiverses, you're all running the experiment 1,000 times. And the proportion of times that you reject the null is either power or type 1 error, depending on what the truth is. So thinking about this, right, this randomness, I want you to think about dice in random sampling. So dices are totally fair, right? At least the standard ones. But it's still possible to roll 5 6s in a row. It's totally still possible, even though it's unlikely because, right, if a dice is fair, you'd expect to get about 1/6 of each outcome over a lot of rolls. So randomness guides our world in some ways. And you have to think about randomness in your trials, right? So randomness guides in medicine and exposures. Not everybody that drinks 10 drinks a day is going to have liver failure. Not everybody that smokes is going to get lung cancer. There's some randomness as far as the processes that work in real life. Weather patterns are random. I mean, if you actually pay attention to the weather and how often they're correct and how often they're not, the truth is they're actually not correct that often, at least to an exactness. Those are randomness things. We try to estimate them based on models, but it's not necessarily a deterministic thing. And human behavior. Imagine we're talking about like predicting a stock price. If people could do that very well, you would have certain people that are totally billionaires and just crushing things. But human behavior is hard to predict, and in a way, it's kind of random, kind of playing off of each other. So goal is to use data to try to sort through things that happen by chance and to find real patterns. So the reason I have this here is I just want to point out that even if you knew that treatment A is better than treatment B, randomly, by running your experiment, you might not observe that in your study. And that's just randomness. Just like with a fair dice, you might get 5 6s in a row. Okay, so now I'm going to talk about an adaptive trial design. So right before, we're enrolling 100 patients in each group. We're observing, we're giving them the treatment A, treatment B. We're observing them, and then we're saying, hey, look, did their tumor shrink? Or did they go into remission? Whatever our outcome is. What if instead of enrolling 100 patients in each group, what if we enrolled 50 patients in each group? We looked at the data and we decided whether or not we should enroll 50 more in each group. So think about this. Maybe after 50 patients in each group, I observe a 70% efficacy rate and a 30% efficacy rate. Well, that might be enough to say treatment A is better already and stop the trial. Why do this? You save money on treatment. You avoid exposing patients to an ineffective drug. If you know that treatment A is better, why are you going to give them treatment B? And it's publishing positive or negative results about the treatment faster, which is going to help more patients in the future, right? It might either encourage people to do follow-up studies to verify what you've seen, or it might discourage people to do that and spend more effort on other potential treatments that might be more beneficial to patients. So imagine this. After we enroll 50 patients in each group, I got a p-value, okay? I've got a p-value, and let's say I'm going to declare A superior, the treatment A superior, if the p-value is less than, this is some threshold. So this is a theta ui, u1. I just want to say it could be, it's some number. Could be 0.05. That would be a p-value less than 0.05. Could be 0.1, whatever it is. This is some number that we're going to figure out based on simulations. We want to set this number to where we don't have a high type 1 error rate, but we do have high power. And we also have another choice. We could declare B superior if this p-value is also less than this threshold and B has higher efficacy. If neither of these happen, we'll enroll 100 more patients. So this is a simple adaptive design. I've got 100 patients enrolled in the 2 treatments. I either stop the trial right there and declare a winner, or I go enroll another 100 patients between the 2 treatments, and then I make my final decision there. Oh, yeah. So if we continue to the full sample size, we have the same decisions. I'll declare A superior if the p-value is less than, this is a different index here. I have U2 because it's the second look. And I'm going to figure out what this value is based on type 1 error constraints and power. What do we want in this? I'm going to declare B superior if the p-value is less than this threshold as well. Or I'm going to say, hey, look, we ran the study. We can't demonstrate that either treatment is better. So that's the decisions here. Now I'm going to set both of these parameters, theta U1 and theta U2, so that the design has good type 1 error and power under different reasonable truths. So as an example, if I do theta U1 equals theta U2 equals 0.025, we're guaranteed a type 1 error of about 0.05 based on Bonferroni's correction. So that's probably something you've also heard about is Bonferroni's correction or Tukey's or things like that. Or I can just simulate the experiment, try out different values, see do I reject the null or not if the null is true, and do this repeatedly. And at the end, I can get a simulated type 1 error rate, and I can get a simulated power under some alternatives. So here I have something called operating characteristics. When you're talking about a clinical trial, whether it's adaptive or just standard, we have these things that we really care about, operating characteristics. Now, it's not just the power, it's the type 1 error, and it's also in adaptive designs, what's your sample size? Okay, so yeah, you have good type 1 error, but are you able to stop the trial early when there really is a treatment difference? So if this is the truth here, under the structure where we enroll 100 patients total, we see do we continue? If we do, we enrolled 100 more. If in truth, it's a 50% efficacy rate in both treatments. The simulated operating characteristics, and this is done from simulation and statistical software, has a type 1 error of about 0.014 and an average sample size of 198.8. That means most of the time it's running to the full sample size when the null is true. If I increase my bounds here, theta U1 and theta U2 from 0.025 to 0.05, I end up getting a type 1 error rate a little bit higher than 0.05 and an average sample size about the same, 0.198, 0.6. And I probably want to calibrate these under a lot of different scenarios. So 50% might seem reasonable. Well, I might look at, okay, how well does this design do if in reality, those 2 treatments have a 60% efficacy rate or a 40% efficacy rate or a 70% efficacy rate. But those numbers that I'm going to try need to be based on like something that's either clinical expertise or literature or observational studies, something like that. So simulating under an alternative here, here's an alternative where in truth, treatment A has an efficacy rate of 50%. Treatment B has an efficacy rate of 30%. So treatment A is better. Then the simulated operating characteristics here with, this is a small cutoff, so 0.025. The power here is 0.711, and your average sample size is 164.8. So that means, right, if you think about what that means, a lot of the trials are stopping at 100 patients, and maybe, I don't know, about 65% are going all the way to 200 patients. And in my simulated trials, 71% are correctly rejecting the null and making the right decision that treatment A is better. If I increase this bound from 0.025 to 0.05, which we saw in the last slide, right, increases the type 1 error just a little bit from 0.014 to 0.0053, but this is still relatively controlled. It's close to what we want. then our power is 0.817. So our power went up by about 10%. And the average sample size is 153. So looking at these two together, if these are reasonable assumptions about how these treatments might actually impact patient outcomes, I'd probably say, hey, look, let's use this more aggressive cutoff of 0.05 because we get higher power and we do get a higher type 1 error rate, but it's constrained to what is usually expected, which is about a 0.05, 5% type 1 error rate is generally what people want. Okay, so right now, as far as now, we've talked about decision rules, right, where we can either stop and declare treatment A better, we can stop and declare treatment B better. What if after we enroll 50 patients in each group, we notice that these two treatments are identical? So 30 out of 50 and 30 out of 50 have efficacy. Should we continue the trial? Should we enroll 100 more patients in this study? When we have demonstrated, at least in the first 100 patients, it doesn't appear that there's any major differences. So what we can do is we can add a new stopping rule, and this is called a futility stopping rule. Why is it called a futility stopping rule? It's basically like, if you continue the trial, it's futile, more or less. Like, you're just going to get information that, hey, look, this thing that we are trying to test to see if it's better, it's not better at all. It's not worse, but it's not better. So you probably want to go with the standard that may have some more historical support among the medical community and things like that. So if we have a p-value that's excessively large, so this is bigger than some parameter that we can change, theta f, then we'll declare neither treatment optimal and we'll stop. So as an example, right, let's say theta f is 0.5, and we use these smaller cutoffs for superiority, 0.025, then we get these operating characteristics. If in truth, pi A equals pi B equals 0.5, so the 50% efficacy rate in each group, we get a type 1 error rate of 0.01, and we get an average sample size of 137.6. I want you to look at before. Under this setting here, without a stopping rule for futility, we ended up going to the maximum sample size most of the time. The average sample size was 198. By adding this futility rule, we've been able to stop the trial at 100 patients and basically not expose patients to something that is really no better, especially if it's potentially more expensive or something like that. Now, in this setting, right, if in reality, the efficacy rate is 50% for treatment A and 30% for treatment B, using this futility rule, we have a power of 0.688 and a sample size of 153.1. So looking at what we had before, we have a small reduction in power, right, from 71% to 68%. So what does that mean? That means about 3% of the trials, when the alternative is true, when the null is false, are stopping early to say, hey, neither treatment is better. But, right, we get this trade-off where if the null is true, we do get this reduction. And I can also move this thing around. Let's say instead of a 0.5, I use a 0.75, okay, instead. Well, in that case, right, I still have good power under the alternative. That's this lower value here. And I get a good type 1 error. But you'll notice, right, my average sample size when the null is true goes up from 137.6 to 161.6. So when you're designing a trial, even in this simple perspective, you need to think about a couple of different parameters, right? You need to think about, well, what should theta U1 be? What should theta U2 be? And what should theta F be? So how do you decide that? You talk to your statistician, you give them some reasonable truths, and your statistician runs with it and does lots of simulations and gives you, hey, look, if we use these cutoff parameters, we get reasonable power and type 1 error and in settings that are actually plausible based on the literature. Okay, so this kind of brings up in general group sequential designs. So what we just did was an adaptive design with one interim look after 100 patients. Remember, at 100 patients, we can say treatment A is better, treatment B is better, or neither is better, stop the trial. Why should we restrict ourselves to sample sizes of 100 or 200? Instead, we can enroll up to 1,000 patients, for example. And every 100 patients that we enroll, randomizing 50-50, we can make those 3 decisions. Well, it's actually 4 decisions, right? 1 is treatment A better? 2 is treatment B better? 3 are neither of these treatments superior? We should stop the trial. And 4th is enroll 100 more patients. So those are 4 decisions that you can make every 100 patients that are enrolled that are driven by data that you have collected. And they are backed up by statistical considerations to make sure that your trial, on average, will operate very well. So there are other adaptive decisions in this context that you could make. You could change your enrollment criteria. So maybe you notice that during the trial, patients with a certain biomarker are doing better in efficacy. Well, you could change your enrollment criteria, and that could be an adaptive decision that you make. You also could adaptively adjust your randomization probability instead of stopping. So, hey, instead of enrolling 50% in this group and 50% in this group, maybe if treatment A looks better, I'm going to enroll 70% in treatment A and 30% in treatment B for the next 100 patients. And that can change that randomization rate. Okay, so I want to take a step back and just kind of talk about, in general, data-driven decision-making and the future. So here I have this little graph right here. This is, I typed into Google and I screenshotted it. Who is the Saints? And you can see that Google has a bunch of things that pop up that autofills. Who's the Saints quarterback? Who's the Saints new head coach? Who's the Saints head coach? New quarterback, offense coordinator, all these things. If you think about what Google's actually doing here is they have thousands and thousands of searches. And they have a lot of those searches probably start with who is the Saints. So they're looking back into their database of all the searches and they're saying, hey, these are the most common things that people typed after who is the Saints. They're using data that they've collected to help you make the right decision about what you want. If you do, I'm feeling lucky at the bottom, it will actually pick the choice for you. So it's giving you here, 10 decisions that you can possibly make based on real data that they've collected, but they could also make it for you. And so from an adaptive design perspective, we have hard bounds, hard rules, which would be based on the data. You either make one of those 4 decisions, stop, say A is better, B is better, neither is better, or continue the trial. Or you can also add some wiggle room there. So you can have kind of these soft decision-making patterns. Okay, hey, you have to pick between the 2 most likely decisions, something like that. And that's kind of the difference here. So data-driven decision-making. So I went to Rice. A lot of my friends went into work for oil. Some of my former MS students are working in oil. So oil and gas used lots of data analysis. They used tons of adaptive decision-making based on data about how do they do futures? How are they doing the drilling? Also, where do they drill? Okay, based on different measures from shale and things like that. Where should they establish new wells? I actually have a friend too that worked on this kind of thing here. So this is a drill at the bottom. And what this drill actually has is like thousands and thousands of sensors on it. And the way that this works is it'll go into the ground, right? And based on whatever the data is, it has statistical rules that make it go left, right, down, further down, up, to try to find where the oil is based on previous data that they've collected. So this is happening in oil, it's happening in tech. It's happening in adaptive, in driving, self-driving cars. So self-driving cars, right, they have a goal. Where do you go? They get real-time sensory data. You can see over here, this picture kind of knows where everything is as far as feeling wind resistance and things like that. Has some kind of camera in front so it can get estimates of how far off the other cars are there. Self-driving cars, that's what they do. They'll make adaptive decisions and they'll not wreck. You have a goal to get from point A to point B, and the cars need to drive themselves. They're going to read in data. In fact, one of the biggest problems that's holding this up right now is they collect so much data on just a 15-minute drive that they have to discard about 40% because they've got thousands of sensors all over these cars. So industry is using data to adaptively make decisions. Why shouldn't medicine? That's my point here of this kind of a bit. So interestingly, the Pfizer COVID-19 trial, to me, at least, I think it's the most famous adaptive design to date. So pharmaceuticals are doing it. They're starting to do it more. So should research. So I had this Twitter post back in September of 2020, because I remember a lot of people were nervous, about rolling out the vaccine. But I looked into the protocol and they had it set up basically as an adaptive design. So I have a link here. And basically what they did, you see here, they've got this quote here. This is from his. It's the total number of patients enrolled in phase 2-3 may vary depending on the incidence of COVID-19 at the time of the enrollment, the true underlying VE, and a potential early stop for efficacy or futility. Those are words that we've used before early in here. So this was a 40,000 person study. It was powered at 90% to detect an efficacy rate of 60%. Now in reality, right, the first Pfizer trials had an efficacy rate of 95%. So we were well overpowered, that study was, to detect a higher efficacy rate than they planned for initially. Now, if I was a statistician on that trial, I wouldn't assume a 95% increase in efficacy. So that's probably why they settled at the 60%. They saw even higher. And that's what they did. They followed patients. They waited until a certain number got sick. Then they looked at their data and they said, hey, can we say that the vaccine is better? If yes, we stop the trial, vaccine's better. If not, is the vaccine not better at all? Is it a futile experiment? If they answered yes, they'd stop the trial. It would be over at that point. If not, they keep going. So I don't know how many looks it took, but I know this trial stopped relatively early. And they were enrolling 40,000 people. Their rules weren't based on, hey, 1,000 at a time. It was more based on how many people are actually getting sick. So I talked about on this Twitter thread, you almost kind of wanted people to get sick, because the faster that people got sick, the faster you could kind of know what works and what doesn't work. Anyways, really, really cool, amazing, famous, and saved a lot of lives. So talking about cancer adaptive clinical trial designs. So we have lots of different ways that this can be implemented in cancer. So dose finding and early exploration, that's one of the important things to do. So you probably heard of phase 1 trials where you choose a dose based on toxicity. You want to find a maximum tolerated dose. So it's basically a dose level with some reasonably high toxicity probability. The reason we have these is because a lot of cancer treatments are inherently toxic, right? You have to kill the cancer, you end up hurting some other organs in your body. Phase 2 could be a different trial. Maybe you've demonstrated safety and you want to pick a dose level for some treatment, chemotherapy, whatever, based on efficacy rates. You can also demonstrate efficacy of a chosen dose on a small scale. That would be the Simon 2S, or you could pick among a set of treatments, maybe 4 or 5 treatments, which one should you go and move on to phase 3 to compare against the standard? And this is an old paper from my advisor. There's another type of trial design as far as dose finding, which is a phase 1-2 trial. That's where you pick a dose level based on both efficacy and toxicity at the same time. So I'm definitely guilty as a big proponent and outspoken proponent of using these phase 1-2 trials, because, right, you want to pick a dose that's both efficacious and not potentially toxic. Whereas in the kind of standard therapy, right, you might pick a dose level that's not too toxic, but if you picked a dose level that was just slightly more toxic, maybe you got a much higher efficacy rate. And I would say, at least my advisor used to say, you know, if you're a patient and you have extra nausea, but your tumor shrinks and goes away, you probably take that. So also changing treatment practice. So phase 3 would be large scale randomized study against some kind of standard, what's being used. And phase 4 would be large scale observational studies after treatment is available. So this is kind of a dose finding paradigm here. Imagine that we have a treatment A. So A is administered at some dose X. This is done before you bring in treatment B. So this is, hey, look, I've got this new treatment. I think this treatment might work. I've got, you know, doses 20 milligrams, 40 milligrams, 80 milligrams, 120 milligrams. What dose should we be giving our patients? So what you do is, here I have a notation here. A of X1 means this is the first dose level here. And at the end of the trial, phase 1 or phase 1-2, I'll pick a dose level. Maybe it's the second dose. And then I'll go on to phase 3 and maybe X2, maybe it's 40 milligrams. In that example, it's the optimal dose for this treatment. And I'll randomize patients between this treatment and the standard. And then in cancer, right, you'll do a study of whether or not the mean survival time here, mu would be the mean survival time. The mean survival time of the dose you picked is equal to the standard versus it being bigger than some threshold delta here. So delta could be 12 months, 14 or 24 months. So 2-year improvement in survival, 1-year improvement in survival. So this is kind of the two conventional strategies for cancer treatment development and evaluation. The first one is you run your phase 1 trial, you pick a dose based on toxicity. So that's this T indicator here. So I pick a dose level, then I go to phase 2. And if this, in a small sample size, demonstrates some increased efficacy, so YE is efficacy, compared to the standard, then you'll go on to phase 3, where you'll compare the treatment that you picked way back in phase 1 to the control therapy C in terms of survival time. YS is survival. Or another kind of conventional strategy is you'll run a phase 1/2 design where you pick a dose that optimizes efficacy rates without being too toxic. And then you'll graduate that to phase 3 and you'll compare that to the standard. So I have this result here, which I don't want to discourage anybody, but it's a true thing that we observe. I don't know, what's happened in the last 10 years, but from 2011, this Aerosmith paper, they say that 50% of phase 3 trials give negative results. So 50% of the trials where you have a new experimental therapy, you've gone through some preliminary stuff. Hey, look, it looks like it's a good treatment compared to the standard. You go on to survival time or a large scale efficacy study. 50% of those trials don't see an actual treatment improvement. And outside of thinking about how these things all connect, I think one big reason for that is there's not enough planning in these early phases. Considering, you could be wrong, right? So I think maybe one of the problems in research is sometimes we're a little bit too optimistic about potential treatment therapies. So having a statistician who can be skeptical, say, hey, look, if we run this kind of study this way, we might potentially graduate a treatment that's not actually any better than what we have. So again, the phase 1 trials, I'll just kind of walk through each part. A phase 1 trial, the goal is to find a dose among a set of doses, X1 to XJ. So I've got capital J doses. So it could be 4 doses, right? Like 20 milligrams, 40 milligrams, 80 milligrams, 120 milligrams, something like that. That has a probability of toxicity. So you have to define what is the toxicity. Is it effusion? Is it fatigue? Whatever that is. Closest to some target probability by some follow-up time. So usually you pick a target probability about 20%, 25%, 30%, or 33%, something like that. It could be higher depending on what your toxicities are. I mean, if it's just general fatigue, maybe you're okay with patients having about a 50% toxicity rate. If your toxicity is a stroke, you're probably only okay with about 5% of your patients having that. And that depends on, right, what the disease we're talking about is, what the cancer looks like, and what kind of efficacy rate you can expect. Patients are usually treated either 1 at a time, 2 at a time, or 3 at a time, and they're followed and observed. Do you have toxicity? So some common designs you may have seen, 3+3 algorithms, which have terrible operating characteristics, which means if you know that the 3rd dose is the best dose, the 3+3 design is going to give you the smallest chance to actually pick that dose as the optimal dose compared to any other toxicity design out there. You also have up and down designs, which is a slight improvement, but still really not beneficial. And then you have designs like the continual reassessment method, where we use data to estimate the toxicity probability at each dose. And we continually assign patients to the dose with the estimated toxicity probability closest to our target. So closest to 30% or 10%. So this is an example, just with my nice little boxes. So I've got, hey, a target of 33% for toxicity rate, and I have 5 doses. Let's assume these are the true toxicity probability. So this goes back to that pi A and pi B, right? I know that this is the truth. I know that the first dose has a toxicity probability of 0.1, which is 10%. Second dose has a 15%. Third dose is 25%. Fourth dose is 38%. And 5th dose is 60%. Ideally, we want to pick dose 4, right? Because dose 4 is the closest to 33%. But the way we'll actually run this is we'll enroll patients at different doses. based on their response rates, their toxicity rates, we'll assign next set of patients to a dose that has the estimated probability closest to what we want. And we'll do that repeatedly until the end. And then we'll pick the dose that we think is the best. What are Bayesian phase 1 clinical trials? So Bayesian phase 1 clinical trials, what we do here is we have a prior assumption about what those toxicity probabilities are. So maybe before the trial starts, I talk to Dr. Ochoa and he says, hey, I think the toxicity probabilities based on literature are 5%, 10%, 15%, 20%, 30%, and 50%, something like that. So what I'll get as far as my estimates of our toxicity probability are going to be a compromise between what we observe in the data and what the prior beliefs are. Now, the reason you might do that is the same reason that you can roll a fair dice and get a 6 5 times in a row. Possibly, right? The third dose, for example, could have the toxicity probability you want, 30% maybe. But it's possible you enroll 3 patients at that dose and all 3 of them get a toxicity just by randomness. It's totally a possible thing that can happen. In Bayesian phase 1 clinical trials, by using that clinical expertise and doing a compromise between these two, what you do is you actually can kind of help guide the trial early on. And later on, that information is kind of washed out by what you actually see in the data. So again, it's different than just looking at the toxicity probabilities themselves, because Bayesian models, they assume the parameters are random, they incorporate prior toxicity probability beliefs, and they're useful in small sample sizes. Phase 1/2 designs, so just a little chat about it. So the most famous design, if you're interested in reading about it, there's a bunch of trials that have used it, is the FTOX design. It's by Thal in 2004. And 463 might not look like a lot, but 463 citations is actually quite a bit as far as statistical methodology. And it chooses a dose level that has ideal efficacy rates and toxicity rates. So you want to find a dose that has a high efficacy rate, but not too high of a toxicity rate. So we worked on this, me and my advisor, in 2020, and we kind of adjusted the way that you quantify what's good here. And what we did was we came up with this scoring system. So here I have, imagine, right? A patient can actually have efficacy and toxicity. So they could have both, right? A toxic event, they have fatigue, whatever it is. They also have tumor remission of their cancer. They could have efficacy and no toxicity. That's a good thing, right? That means that the tumor is shrunk or is in remission and you didn't feel terrible from the drug. You could have toxicity and no efficacy, which means they felt terrible and their tumor didn't respond to treatment, and you could have neither. So what we did is, and thought through this, is you elicit scores for each of these outcomes. So the score for here, I have efficacy, no and yes, toxicity on the top, efficacies on the left-hand side. So in this case, a yes, no, a yes efficacy, a no toxicity, that's the best thing that can happen. So we'll give that outcome a score of 100. A yes toxicity and no efficacy, that's the worst thing that can happen in the trial. So we'll give that a score of 0. Then what I'll do is I'll talk to clinicians. Okay, well, thinking about what the drug is and what the disease is itself, is a no-no the worst thing, right? So maybe there's some salvage therapy that they can have, and a no-no, so no efficacy and no toxicity, might not be the worst thing because there's other sort of salvage treatments that they could have. Or it could be a terrible thing. Maybe there's no other alternative here and you really want to have efficacy. In the latter case, you'd put a lower score, closer to 0. In the former case, you'd put a little bit higher score. This yes case, right? So this is another thing to elicit from clinicians, scoring this, giving me a score for this. Okay, if efficacy is tumor remission, so the tumor's gone, but toxicity is stroke, you probably don't want this outcome to happen a lot. If your toxicity is fatigue or nausea, maybe this score could be higher or close to 100. And so that comes between thinking about what can actually happen to these patients. And then you'll run simulated trials based on these scoring systems. And based on reasonable things, I'll give you, hey, what's the probability we pick the right dose? There's many published versions of this type of trial. So here's an example. Okay, so I have these scores. And in the end, what I want to compute is what is the average score, or what's the expected score for each dose? So if I look at this, right, I have dose 1 has a a modeled score. So this comes from statistical models. It's magic. And what you provided me as a clinician. Dose 2 has a score of 20. Dose 3 has an estimated score of 45. Dose 4 has an estimated score of 65. And dose 5 has an estimated score of 12. So the reason I like to think about these is you can say, hey, look, the score of dose 4 is the highest. It's the highest by about 20. So I'd assign the next set of patients to dose 4. And if the trial is over, I'd say, hey, look, dose 4 is the best. Let's go ahead to the next phase of testing. Likewise, here's another sort of set of examples down here. Here, I enroll more patients, I get more data, and maybe now I see that dose 1 is actually the best. Dose 1 has a score of 75, dose 2 has an estimated score of 60, 3 is an estimated score of 45, 4 is an estimated score of 25, and 5 is 12. Then I'd assign the next set of patients to dose 1, since that is the highest score. If the trial is over, I'll declare dose 1 the best. So in the Pfizer trial, they talked about it. The phase 2 part of their trial was establishing dosing in teens. And I don't know what exactly their structure was, but they could have done something similar to this, using some kind of scoring system, estimating these scores for each dose, and then sequentially assigning teens to those doses based on the outcomes that you already observed. Okay, you also have phase 3 trials in cancer vaccines. So it's a fully randomized trial. You need to demonstrate evidence of comparative success against standard of care. So A at chosen dose versus treatment B. Tracks patients over time and it decides when the event happens. So the event could be death, right? So in cancer, you might follow people until they have death, until their cancer progresses, or some other thing. It's a time to event thing. In the Pfizer COVID-19 trial, the event was just whether or not you got COVID, the time that you got COVID. And this deals with censoring, which is accounted for in data analysis. So what is censoring? Censoring is basically, let's say I'm following, some patient and they have a very aggressive cancer, and I'm trying to follow them to see their survival time. So maybe after a year, this patient hasn't died, but I want to make a data-driven decision at this point in time in the trial. Well, I know this patient hasn't died by 1 year. I know that they will die sometime in the future, because we all will, whether or not it's cancer. But I actually use that partial information. Hey, look, they've lived a year, they're still alive, in making decisions here. So the way that these work in cancer and in vaccine trials is after a preset number of events, think the NA and NB in the simple example, we stop and we'll say, hey, is treatment A better? Is treatment B better? Or we'll say, hey, look, we're going to stop for futility, or we're going to keep randomizing patients between the two treatments. Some other interesting designs that are coming up are platform trials. So this is the I-SPY 2 trial. You could look it up if you're interested. And basically what this trial does is they've set this up, I don't know, maybe about 10 years ago. It's Donald Berry's group, and they work with different researchers, but more or less, right? Imagine a situation where instead of like 5 preset treatments, maybe you're running a trial of 5 treatments, and 2 years down the road, while you're still running this trial, it looks like, hey, this new 6th treatment might be better than the other 5 that we have. Well, that new 6th treatment could go replace one of these. You can drop treatments as you go along. It's basically sorting through multiple treatments over time and making decisions about, hey, should we go ahead to a phase 3 trial? Should we do whatever? It mix and matches rules too. You can have lots of different stopping rules, but it's data driven. Do you graduate a treatment? Do you stop treating patients at a treatment? And I've seen them talk about this trial before, different conferences, and they've talked about the treatment arms that have been dropped, the treatment arms that are still running through, and they're still adding and dropping treatment arms sequentially to this day. Another area of research that I think is interesting as far as adaptive designs is N of 1 trials. So these are really popular in Australia. I've worked with some people there to look at simulation-based studies of their power. And basically what happens is the patients serve as their control and their treatment group. So you might be familiar with a crossover design. This is a crossover design with multiple crossovers. So sequentially what you do is you'll treat the patient, you'll observe your outcome, you'll wait for your treatment to, quote, wash out, you'll give a patient a control, you'll observe their outcome, you'll wait for a treatment to wash out, and you'll repeat that over and over again. So as an example, right, maybe I have one type of vitamin that I want to give somebody versus another. Okay, so I could give them the vitamin for 2 weeks. At the end of their 2 weeks, maybe I measure their blood pressure or something like that. And then I give them 2 weeks off of the vitamin. Then I give them the second vitamin, wait 2 weeks, measure their blood pressure, and I repeat that process over and over again. So what this does is it actually gives us better power overall to say which treatment is better, because now instead of, for example, 30 patients, what I really have is 30 patients times however many times I cycled them through them in terms of observations. Basically, each patient really counts for 5 or 6 observations. And it also allows individualized decisions, so personalized medicine. So I can tell patient 1, hey, look, vitamin A works better for you. Maybe it doesn't work better for everybody else, but the data that I've collected on you says that you should be taking vitamin A instead of vitamin B. Dynamic treatment regimes. So this is another adaptive decision-making thing. And this really actually, I think, mimics the way that, and you can tell me if I'm wrong, the way that clinicians think about treatment. So a dynamic treatment regime, I've got a link here, consists of a sequence of decision rules, one per each stage of the intervention, that dictate how to individualize treatments to patients based on evolving treatment and covariate history. So here's an example. You give alcohol dependence naltrexone. Do they relapse and have heavy drinking, 2 heavy drinking days in a row? If yes, you'll start cognitive behavioral therapy. If not, you'll continue the treatment. So this is super common in cancer and tumor response. So, hey, I'm going to try this chemotherapy. Oh, it doesn't work. I'll try radiation, that kind of thing. So how do you optimize the way that you're going to treat patients through these sequential decisions that you're going to make? So here's just an example looking at weight loss. So here I have IBT as individual behavioral treatment, MR is meal replacements, ACT is acceptance and commitment therapy. So as an example, right, I've got a patient, they're overweight or obese adult. If they have no history of emotional eating, I'll give them IBT for 5 weeks. If they have respond at the end of week 5, so they do decrease in weight, I'll continue IBT. If they don't respond by the end of week 5, maybe I'll do IBT and I'll do meal replacements. Likewise, if I don't have a patient with a history of emotional eating, I'll do IBT for 10 weeks. If they respond at the end of 10 weeks, they lose weight, I'll continue the therapy. If they don't respond at the end of 10 weeks, I might switch to this ACT here. Now, how did I decide that this was the optimal way to do this? You use SMART designs. So SMART designs, also Peter Thal is a big pioneer in it. And the way that you do this here is R is randomized. I'll take my patients, I'll randomize them to these 2. And then I'll wait. Okay, did they respond? If they responded, I'll continue. If they didn't respond, maybe I'll randomize them to ACT or IBT and MR. So I've got, right, 2 decisions. One is they got IBT. The second one is one of these 2 here based on their response. Likewise, in the second group, for the non-responders, I might also randomize them to the 2 treatments. So it's just one example, and it can be based on patient covariates as well, not just their outcome. Enrichment designs, this is a growing area. So it's an adaptive thing where you adaptively change your enrollment criteria. You're trying to find a subset of patients that most benefit from some treatment. And it's based on covariates or medical history. So it could be based on comorbidities or age or biomarkers through sequencing. And what you want to do is you want to figure out, hey, what patients actually respond to this treatment the best? And you adaptively refine the subset of patients where they benefit based on the data. So I've had personalized medicine here. I think, we'd like to move in that perspective where somebody comes in and they get the best treatment for them. This is kind of hinting at that. If somebody has qualifications that based on their data or covariates, a history based on their data, looks like they'll actually benefit from this treatment, you give them the treatment. And so we're trying to figure out who benefits from those treatments in enrichment designs. Subgroup specific designs. So this is a big area too, similar to enrichment designs, but with pre-specified subgroups. So it could be based on cancer stage, biomarker status, age group. You could do phase 1 trials. So you could do dose finding. You can borrow strength from the different groups. You can do a phase 1-2 trial. You can do a phase 3 trial with decision-making within each subgroup while borrowing strength. Well, I've actually done a lot of work in this area. And I think that this would be a really promising thing. One of the trials actually was used at Dana-Farber Cancer Center. So I worked with the researchers there to develop a dose finding trial. They were trying to find the dose level out of 4 doses that had some toxicity probability closest to some target. And this was in patients with pediatric brain tumors. So they were using the same doses in each set of patients as far as broken down by BMI. So it was like 20 milligrams per however many pounds, something like that. But they thought that maybe teenagers, because they're, I guess, going through puberty, might respond differently to the medication than children. So they had 2 different subgroups and they did dose finding within each of those. The trial is actually done. I haven't seen what the results come out yet, but that's an example of dose finding within 2 groups at the same time to make a decision about what's optimal. Ordinal outcomes. So we don't have to just think, and this is that trial that Dr. Ochoa talked about at the beginning. So recently we've moved to discussing ordinal outcomes in COVID. So using scoring. So remember this, scoring right back here, had these scores. So we can score ordinal outcomes as well. So in COVID, right, somebody could die. They could be on ECMO or invasive mechanical ventilation. And that's probably the worst thing that can happen to a patient. So they get a score of 0, right? Because I want to find the treatment that has the highest estimated score. They could be hospitalized on high flow oxygen therapy. The researchers I worked with said this was the second worst thing. They gave it a score of 35. They could have hospitalization with other supplemental oxygen, said this was the 3rd worst thing with a score of 65. They could be discharged, so back at home with supplemental oxygen. They gave it a score of 75, and they could be hospitalized without supplemental oxygen, in the score of 80. And the best outcome for a patient as far as being hospitalized with COVID is they leave the hospital and they don't have supplemental O2. And this was also very early on in COVID when we were thinking about it. So, you can imagine now there probably would be long COVID would be part of this. So this trial was supposed to run at LSU to pick among 3 treatments. We were looking at azithromycin, or hydroxychloroquine and azithromycin, and also the placebo. It started running, and then after sort of the FDA guidance on hydroxychloroquine, the trial was shut down. But we got a nice paper out of it and developed software to actually run this kind of trial. So if you can think of something where you have multiple treatments, more than 2 treatments that you want to test what's better, you might think of, and you have a complex ordinal outcome, you might think about scoring this thing. And the nice thing about what this trial did, which is unfortunate that we didn't get to run it, is we had sequential rules to drop treatments that were worse than the others, to stop the trial and declare one treatment superior, and also to stop the trial and declare no treatments are better. And if you check out this paper via simulation, show that it works really well compared to some other ways to approach this. So using other outcomes, you could think about designs based on longitudinal continuous outcomes. So for example, how does longitudinal BMI trend based on weight loss medication? How is longitudinal A1C in patients with HIV? And thinking about making decisions based on control of those, or glycemic control in diabetic patients. You can also do designs based on multiple different types of events. So different types of toxicities. You could have more than one group. You could have a mild toxicity and severe toxicity, headache versus a coma. Maybe you want to control for both. Maybe you want the probability of a headache to be, you're okay with about 40–50%, but you want your probability of coma to be about 5%. So you can dose find or make decisions about treatments based on different criteria there. Also tumor reduction, overall prognostic benefit, other things. Designs based on multiple transition times. So in reality, cancer is a complicated thing, right? You could have your cancer progress, that could get then reduce, then go into remission, and then you have recurrence, and then you die, or your recurrence goes into remission again. You really have a disease, right, where you have time to event information on when people are going from a remission to a recurrence, or going from recurrence to death, and things like that. Also time till surgery, when the second surgery is needed, things like that. So I just want to kind of plug this, got 20 minutes left, just quickly plug kind of phase 1, 2, 3 designs and the whole process. The goal here was to get through this problem that 50% of phase 3 trials fail. So we're thinking ahead about how the data informs the decisions. And it basically puts phase 1/2, which is dose finding, and phase 3, which is it better than the control, together in case you make the wrong decision in phase 1/2, right? Because possibly a dose that you pick in phase 1/2 based on 60 patients isn't really the dose level that's going to improve patient survival the most. In our study, we showed that this framework that I'm going to talk about increases the probability of finding a new treatment benefit from 10 to 80%. It's pretty substantial. And not that I say that, this is the thing that we should be using all the time, but I'd like to see adaptive decision making be thought about in this way through the whole drug development process. So as an example, right, this is how it works. We do a phase 1/2 trial, we pick a dose level, then we start phase 3 by randomizing patients between our treatment and the control until we see some number of deaths. Okay, so this is a parameter that I set here. So it's a simulation thing. I do simulations to see, hey, are we getting good type 1 error? Are we getting good power? Then at the end of stage 2, I'll possibly re-optimize the dose. I'll pick a new dose to use the remainder of the trial. And that's based on efficacy, toxicity, and long-term survival of patients treated with A in stage 1/2 and in this randomization. And then I'll finish out my study doing a group sequential design, comparing the dose I re-optimized to. I could pick the same dose versus the control. And this is an important idea. I mean, it was important enough that it won paper of the year in biometrics. And again, that's a very prestigious statistical journal, because the idea here is something that I think could be really, really useful in the future for doing medicine advancement and dose finding. So just graphically how it works, imagine in phase 1/2, we pick dose 4. Okay, we pick dose 4 and we're starting dose 4 against B. And then based on it, the data suggests actually dose 3 is the best. Okay, well then I'm going to switch patients from dose 4 to dose 3. I'm going to discard patients treated at dose 4. And then I'm going to do a randomized comparison of the experimental agent at dose 3 compared to the control, and a group sequential design. So again, the group sequential is I could stop after any certain number of patients and declare one treatment better or keep going. So this is 2 different adaptive decision-making structures. 3, actually. One is, hey, what dose do you pick here? Second one is, do you re-optimize? And the third is group sequential designs. Lots of adaptive decision-making based on data. Here's another example. Let's say at the end of phase 1/2, I actually picked the right dose and I start my trial, and the data suggests that this dose has the best mean survival time. And then I'll continue and I'll do a group sequential comparison. This is just a nice graph. We had a response to the paper, and Zal et al. Made this really nice graph to kind of explain. So in phase 1/2, you can see patients are getting treated at the first dose, they're getting treated at the second dose, they're getting treated at third dose, fourth dose. And what we do in this is we just continue to follow them. So maybe you're looking at efficacy within 30 days. You could still follow that person and see how long that they live afterwards. So you could see that, these people, right, this person, for example, is living all the way into the phase 3 part of the trial. And this is the dose I picked maybe, so I start the trial, and then I re-optimize to dose 3 and do my randomized comparison. So this is just these survival times through this whole process. I really, really like this graphic. The future, it's bright. Actually, right now, there are thousands of adaptive clinical trial designs out there. They're all statistical in nature. I think the problem is a lot of them haven't been used. But it shows that there's not a lack of creativity in developing these tools. To me, I think the best way to make a new adaptive design is to talk to clinicians and see what they want and what are the problems and challenges that they could face. Then the statistician has to come up with, hey, a design, a decision-making procedure based on data that gives you good type 1 error, it gives you good power, and it gives you, decreased sample sizes in adaptive decision-making. So if you meet with a statistician, you explain your idea carefully, they could construct a new trial just for you. Algorithmic decision-making might help patient enrollment as well due to increased trust. If you're a patient and you know that, hey, I'm going to enroll in this trial, it's not up to the doctor what treatment I'm going to get, how they're going to treat me. It's based on the data, the observed and evolving data. You might have a little more trust in the process as well. So just to say too, like you can develop new designs based on needs. And what I'd like to do here is really to contribute more to that, to actually doing designs that are going to be used. And I think a lot of these trials maybe are just kind of like exercises. We want to develop tools that are going to be used to help make decision making. Again, work with a statistician. They can help you collect the data you need. They can help you plan out your sample sizes, establish protocol. There are new statistical methods coming out every day. There's like 20 or 30 really high profile statistical journals, and they have a journal published every month that has new statistical methods. So there's a quote about asking a statistician here for help after the data is collected. And he can perhaps say what the experiment died of. That's it. He can't really help you at that point. So I think working closely with somebody that understands data and how you maybe make decisions as far as research goes and treatment benefit early on can really help avoid issues where you have 50% of phase 3 trials failing. And that's it.
Dr. Chappell, thank you. Thank you so much. That was just so full of valuable information. I have a number of questions, but I want to defer to, first of all, our WIN CIPP students who are working with Drs. Ochoa and Dr. Mile. If you have questions, and of course we also have some colleagues of Dr. Ochoa and Mile on the call. Thank you all so much for joining today. But if we could allow students maybe to have the first go at questions, and then others can join in, that would be great.
Let me ask Dr. Chappell to stop sharing his slides so we can see the, there you go. Thank you.
Thank you. And please feel free to just unmute yourself and begin talking. If we get a lot of crosstalk, then maybe we'll go to raising hands. But students, do you have questions or thoughts? I'm giving you a chance because when the rest of us get going, it might be harder to get in.
Maybe they're just shy because they...
Yeah. But this is really great. Well, then I'll open the floor for questions, comments. I see something in the chat. Is that a question? Oh, that Casey says thank you, as does Yassine Khan. Thank you for a super informative session. Are those comments?
I have a question.
Yeah.
So I actually have more than one, but let me start with the, if these types of trial designs save time, space, funding, all of the above, why are they not used more frequently? Is it because people don't understand them, because they're so new, or because we're stuck in the old models of having to do thousands of patients in order to draw any conclusion of this?
I think a mix of all of those. Definitely, they really haven't been around since, I think the Simon design was the first, and that's 1988. So, if you think about even just in treatments that actually have shown benefit, how long does it actually take to get to patients? Mm-hmm. So that's part of it. I think another part of it is statisticians are trained in theory, and maybe the communication's not quite there as much. So I think part of how do you disseminate it in a way that's understandable without, I think, as a field, sometimes we get a little bit high and mighty. Oh, you're doing the wrong thing. But in reality, the only reason we have a statistical field is to actually help the scientists, do research. Right. So I think that part is getting kind of lost of it. And then it's a little bit of an extra hurdle, right? So imagine, right, we're doing a dose finding trial. Okay, you've got now your 3 patients you want to enroll at the next dose. You have to send me the data. I have to make the decision and communicate with you. Okay, enroll the next patient at this dose. Now, it's not necessarily something that takes a long time to do, but there is that kind of extra level of we have to have a communication about the next step based on the data, and then the decision is made.
Right.
So I think a lot of those things will play into it together.
but it is clear, for example, that the speed of approval of drugs today in cancer is, manyfold higher. I mean, in the past, up to the early 2000s, I think they used to approve somewhere between 3 and 4 drugs a year of these very long and extensive and complex clinical trials, while today they're approving drugs with 10 patients, 1 patient, a few patients here and there, because in part because the results are so dramatic that it becomes, I wouldn't say irrelevant, but it's not possible to just do randomized trials when those results are so significant. Let me just ask another question. So I think this, to me, makes me, and especially for our young clinicians, and I see Dr. Castellanos here with us, I think we need to start thinking about these kinds of structure for the trials, because clearly in a smaller cancer center like ours compared to the big ones, I think we could use the economy of patience and funds and effort to put together some very interesting and important clinical studies.
Yeah, and Dr. Chappell is going to meet with me next week because we're going to maybe start designing a trial to do here. So yeah, it's exciting to have such an expert to collaborate with.
Fantastic.
Absolutely.
There is a question from a student. Lynn Cayley wants to know, what made you choose this area of research?
So what made me choose statistics was, in general, was Freakonomics, the book. And whenever I started, I didn't quite understand, at least, that when you're doing statistical research, it's a little more theoretical and less like data driven. I mean, that's a lot of my job now, is the data part of it. But then really I went to a talk at MD Anderson, and there was a famous speaker there. And at the end of the slide, they had my advisor there. And he's got like his handle is rex@mdanderson.org, because he likes dogs, but he doesn't have a dog. And I went and talked to him, and that's really how I got into it. I fell into it by reaching out to somebody that was an expert in the field. It sounded interesting, and he taught me everything I knew up until 4 years ago. That's pretty much my story.
In other words, a good mentor makes a difference. I like to add a little bit to what Dr. Ochoa was saying. The assumption behind the older clinical trial designs is that patients are more or less comparable. So if you take a large group of patients and you divide them into 2 arms or more that receive different treatments, the only difference in response will be due to the treatment. The biology says that that's not true. Patients are not the same as one another, and the more we learn about the biology of humans, the more we understand that we have to tailor treatment. That's the whole idea of precision medicine. And with these clinical trials, one can do precision medicine in a rigorous fashion.
I totally agree with that. And to handle that well, you have to think about, okay, well, what patient covariates could actually impact not just how they receive the treatment, but how they not necessarily respond to treatment, but how their outcome would change regardless of treatment. And then we work with a statistician and think about, well, how do we model this? How do we set up a theoretical model that the data is going to infer? And then how do we use our estimated model to actually make decisions that are good? All of that comes together, and it's a collaborative process.
Nice.
Can you use, I mean, obviously we're thinking of patients because that is the ultimate goal of the research we do. But we also spend a significant amount of time, energy, and cost on doing clinical studies in mice. And we always have the traditional model of the control versus the treatment 1 versus the treatment 1+2 versus each one of them separately, all that kind of stuff. And, you know, once you start adding up, each one of those experiments will cost, you know, $10,000, $15,000 each. If we were to use a plan to design these adaptive studies in mice themselves, we might be able to get some good answers, at least in these initial studies, while we get to the more detailed studies on molecular mechanisms and things of that sort. Is that
100%.
oh, really? Are people using that kind?
Oh, I don't know. I don't know if people are, but 100%, anything that applies to people will apply to mice in this context as well, with the, you know, ethical advantage of not being people. So that could sort of change the way you have your rules as well.
Except the tails.
Say that again?
Except the tails.
Yeah.
Humans have
Well,
short.
I think it would be, you know, believe it or not, we do spend a tremendous amount of money doing these animal experiments. And we might be able to use this type of design in some of the experiments we do, and maybe we should have closer conversation with some of our investigators, because that might help us either save on what we need to do or move them quicker, or both.
Absolutely. And just as an example, one thing that one can do in cancer trials in particular is use organoids from the tumors of patients to determine whether or not these respond to your planned treatments, and change how you're going to design your trial based on it. You can quickly generate organoids from a sample of tumor. You don't need to put them in mice. They will respond to all sorts of things, not only drugs, but immunotherapy as well. And if, let's say, organoids from 10 patients respond to a certain thing and from 20 patients don't, you have some idea of how to make those decisions, how to assign those scores. And then you can use that in your meeting with a biostatistician to say, you know, it looks like 2/3 of the patients are not really going to respond to this if our organoid data are correct.
Totally. And even playing off of that, so I don't know that this exists or not. Like I said, there's lots and lots of trials, but you could even sort of play with modeling the correlation between those organelles response and the people's response. You have more information from the organelles, less information from the people, and make decision making based on that whole structure. But I think understanding how these things might play, then you set up the statistical model, then you collect the data to actually estimate the model, then you make the decisions. And we're going back to mice. So I know that something that people use a lot is they'll adjust sort of their prior beliefs based on historical controls. And so they might rely more on the historical controls, less on it, and more on the current data, things like that. So you could imagine mice kind of playing into that perspective as well.
Can these types of statistical models be applied using retrospective samples? Because what we saw was mostly prospective design of clinical trials and things of that sort. But we also have the fortune of being able to access archival tissues and things of that sort. Can similar models as what you presented be used retrospectively to study things that we find in archival tissues?
Yeah, absolutely. And even I'll go even further than that. You could even, if you know when each of these outcomes are taking place, you could even go back and say, hey, look, we have the data, we collected it. Let's say we did an adaptive design instead. When would the trial have stopped?
Yeah.
You can figure that out retrospectively, which might give enough to say, hey, look, you know, we spent $100,000 more than we needed to on this study.
Right.
Maybe going forward, we should do an adaptive approach.
Very interesting. Really very, very interesting.
So we are at time. This is really, you can tell there are many questions. I have some of my own. Really thought-provoking, very interesting discussion, Dr. Chappell. Thank you so very much for being with us, for sharing your time and your expertise. Thank you, Dr. Ochoa, again for bringing Dr. Chappell to us. And thanks everyone for being on this call today. Just always a pleasure to be with you all. And for those of you who will be at the symposium this weekend, looking forward to seeing you in person. Dr. Chappell, if you are interested in joining us, by all means, you are invited. It's going to be a good time. So we'll have all students from all 5 cities from all over the country convening over at Loyola right down the street. So
On Sunday, correct?
on Sunday, starting at noon on Sunday until 8 p.m. and then beginning at 8 a.m. on Monday until 3:15. So please do join us if you'd like to come in for those discussions. There will be further discussions, deeper dives into community-based participatory research and professional development sessions, lots of great conversations. And Dr. and Mile and Castellano and Morel are going to be on our panels. It's going to be a lot of fun. So
Send me the information. If I can come, I will for sure.
I will definitely share that information with you. But
Well,
thank you again
for
so
having
much.
me. I enjoyed
Thank you.
talking to you, and I hope that everybody starts thinking about these things going forward. I'd like to say that 50% figure goes down to 10%, in
Yes.
20 years. So that's the goal. And thanks everybody for coming.
Sit down with the statistician at the front end of the study.
100%.
Thank you. Thank you, Dr. Chappell.
Thank you very much. Everyone have a great afternoon.
Bye-bye.